Today we're going to be talking about mitosis. The process of mitosis is fast and highly complex. The sequence of events is divided into stages corresponding to the completion of one set of activities and the start of the next. Let's look at these stages. The mitotic phase is a relatively short period of the cell cycle. It alternates with the much longer interphase, where the cell prepares itself for cell division. Interphase is divided into three phases: a first gap, a synthesis, and a second gap. During all three phases, the cell grows by producing proteins and cytoplasmic organelles. However, chromosomes are replicated only during the synthesis phase. All these phases in the interphase are highly regulated, mainly via proteins. The phases follow one another in strict order, and there are checkpoints that give the cell the cues to proceed from one phase to another. Altogether, interphase takes up roughly 90% of a cell's lifespan. At the onset of prophase, chromatin fibers become tightly coiled, condensing into discrete chromosomes. Since the genetic material has already been duplicated earlier in the synthesis phase, the replicated chromosomes have two sister chromatids bound together at the centromere by the cohesive protein complex. Chromosomes are typically visible at high magnification through a light microscope. Inside the nucleus, the nucleolus in the nucleus disappears from view. This is noteworthy because the cell does not need to divide the nucleolus right away. It will later reform when the nucleus divides completely. During metaphase, microtubules find and connect kinetochores in prometaphase. Then the two centrosomes start pulling the chromosomes through their attached centromeres towards the two ends of the cell. As a result, the chromosomes come under longitudinal tension from the two ends of the cell. The centromeres of the chromosomes, in some sense, convene along the metaphase plate or equatorial plane, an imaginary line that is equidistant from the two centrosome poles. This line is called the spindle equator. This even alignment is due to the counterbalance of the pulling powers generated by the opposing kinetochores, analogous to a tug-of-war between people of equal strength. In certain types of cells, chromosomes do not line up at the metaphase plate and instead move back and forth between the poles randomly, only roughly lining up along the midline. When every kinetochore is attached to a cluster of microtubules and the chromosomes have lined up along the metaphase plate, the cell proceeds to anaphase. Two events then occur. First, the proteins that bind sister chromatids together are cleaved. These sister chromatids now become separate daughter chromosomes and are pulled apart by shortening kinetochore microtubules and move towards the respective centrosomes to which they are attached. Next, the polar microtubules elongate, pulling the centrosomes apart to opposite ends of the cell. The force that causes the centrosomes to move towards the end of the cells is still unknown, although there is a theory that suggests that the rapid assembly and breakdown of microtubules may cause this movement. At the end of the anaphase phase, the kinetochore microtubules degrade. Telophase is a reversal of the prophase and prometaphase events. It cleans up the after effects of mitosis. At telophase, the polar microtubules continue to lengthen, elongating the cell even more. Corresponding daughter chromosomes attach at the opposite ends of the cell. A new nuclear membrane, using the membrane vesicles of the parent cell's old nuclear membrane, forms around each of the daughter chromosomes. The nucleoli reappear too. Both sets of chromosomes, now surrounded by new nuclei, begin to relax or decondense back into chromatid. Mitosis is complete, but cell division is not yet complete. Cell division is complete when the separate process of cytokinesis begins, which begins at the same time as telophase.